Macrophage Systems Biology by Brian Geniuson, research scientist. This short examines how macrophages function as central regulators of immune response through integrated signaling, cellular activity, and system-level interaction. Immune activation begins with pathogen recognition, where macrophages detect external threats through receptors, including toll-like receptors and Fc receptors. This recognition triggers intracellular signaling pathways, primarily nuclear factor kappa B and mitogen-activated protein kinase pathways, initiating a coordinated cellular response. These signaling cascades drive cytokine production, including tumor necrosis factor and interleukin 6, which act as key mediators of inflammation and immune communication. At the cellular level, macrophages perform phagocytosis, engulfing and processing pathogens through phagocytic mechanisms that enable antigen presentation and downstream immune activation. Macrophages do not function in isolation. Through cytokine signaling and direct interaction, they communicate with T cells and the endothelium, forming a crosstalk network that propagates immune signals across tissues and vascular systems. This network includes feedback loops that amplify or regulate inflammatory responses depending on physiological context. When dysregulated, macrophage activity contributes to pathological conditions including chronic inflammation, atherosclerosis, and tissue damage. These outcomes reflect sustained inflammatory signaling and impaired resolution mechanisms within the cellular network. From a systems perspective, macrophages act as both sensors and effectors, linking molecular detection to tissue-level and system-level outcomes. This establishes a core principle of systems biology. Changes in cellular behavior drive changes in system behavior. This content is provided for educational and scientific purposes only and does not constitute medical advice.